Okey, boleh nampak slide saya?
Boleh doktor?
Boleh doktor.
Okay. Alright, thank you for the response. Okay. Dah sikit dengar. Ingatkan baru nak panggil nama seorang-seorang. Just nak make sure you all ada ke tak. Okay. Alright. Okay. So we start at this slide. Okay, on the formation of AMP and GMP from inosinate or inosine monophosphate. Okay. Please excuse. I have a very poor son. Okay, dengan saya ni. That's why I cannot come today. I'm just worried. Saya risaulah ni sebab maybe he will get admitted ke apa ke, but we'll see. Okay, that's why I have to tak boleh nak ke UPM. I have to stay with him while waiting for the because we have the appointment, hospital appointment this afternoon. Okay, sorry for that. Okay, so we stop at this slide. Okay, you should know from 11 steps. So this is the product, okay, IMP, okay, inosinate or inosine monophosphate. So we know in the purine biosynthesis, we like to produce AMP and GMP, okay. We know that adenine and guanine is part of is classified as a purine. It has this structure. Okay, as I mentioned last week, you should be able to differentiate the structure of the nucleotide. Okay, kat mana apa beza adenylate and guanylate? Okay, is it in terms of the functional group? Okay, maybe in terms of the location of certain molecule. Okay, you should be able to identify that. Okay, so we know that from IMP, it can undergo another two steps, two reactions to produce adenylate. Okay, and which in the production of AMP, it will produce this adenylyl succinate. Okay, and it involves the hydrolysis of GTP. Okay, so it's a high energy reaction. Okay, that catalyzed by adenylyl succinate synthetase, and this IMP is react with aspartate to form this adenylyl succinate. Then adenylyl succinate undergo it release fumarate, okay, that catalyzed by adenylyl succinate layers to produce AMP. Okay, same goes to the production of GMP from IMP. It involves two steps, two reactions. Okay, one is the first reaction catalyzed by MPP hydrogenase that involve hydrolysis process. Okay, it involves is an oxidation reaction that produce xanthylate, which is XMP. Okay, then XMP it react with glutamine, that which glutamine act as a amino group donor. Okay, and it involves ATP. So it's energy catalyzed reaction to form guanylate. So there is an addition of amino group, okay, to xanthylate to form GMP. Okay, so I hope at this slide you can understand the process, the 11 steps that produce IMP. So from IMP, so IMP is the key metabolite to get this AMP and GMP. Okay, because purine is adenine and guanine. So how to get AMP and GMP? It need to undergo the 11 steps. So from IMP, once we get IMP, it can be converted to AMP and GMP. Okay. All right. So if you compare, okay, that one is the de novo pathway for purine. Okay. Now let's look. Okay, what is the difference of the de novo pathway for pyrimidine nucleotide? So pyrimidine, kita tahu it consists of uracil and cytosine. Okay, uracil or cytosine or uracil or thymine. Okay. All right. So this is the structure of uracil. Okay, this one is for uracil and this one is for cytosine. Okay, apa beza? Cytosine, it has this amino group. Okay, instead of uracil, it has this keto group. Okay, keto group. Okay, so this is structure of UMP. Okay, uridine O or uridine monophosphate. Okay, and this one is CMP, cytidine monophosphate or cytidylate. Okay. So what are the difference between the pathway, the biosynthesis of purine and also biosynthesis of meroperidine? So we have discussed. Okay, you already know what are the sources for the purine structure. Okay, kita ada fumarate, we have aspartate, we have glycine, we have glutamine, we have bicarbonate. Kan? Okay, ada a few. Okay, but for pyrimidine nucleotide, the sources comes from, okay, this aspartate, PRPP, and also carbamyl phosphate. Okay, only three. Okay, tiga je. All right. Okay. Okay. If you still remember in the biosynthesis of purine, okay, in the pyrimidine biosynthesis, before it synthesizes the 6-membered pyrimidine ring, okay, before it can be attached to the ribose 5-phosphate base dengan purine biosynthesis. Okay, and what makes this biosynthesis special? Because it requires carbamyl phosphate. Okay, if you remember your urea cycle, carbamyl phosphate is one of the intermediate that involved in the urea cycle. Okay, so this is a very important intermediate that involved in the pyrimidine nucleotide, in the pyrimidine nucleotide biosynthesis. Okay. Okay, so in general, okay, the process of pyrimidine biosynthesis is much simpler. Okay, it's much simpler than purines. Okay, it is very simple. Okay, so what we can say that so it involves okay, aspartate, PRPP, and also carbamyl phosphate. Okay, if we look, okay, if we look at the structure of purines, kalau you okay, all right, okay, saya nak share this structure. This is adenine, okay, purine, I'm looking for structure of pyrimidine. Okay, so this is structure of pyrimidine. Okay, so if you look at the structure of pyrimidine, contoh cytosine. Okay, we know that. Okay, N1, C4. Okay, C5, C6. Okay, alright, apa lagi? Okay, C2 is rises from bicarbonate. Okay, okay, and 3 probably contributed by glutamine. Okay, and amino. Okay, all right. All this carbon C4, C5, C6 is derived from aspartate. Okay, C4, C5, C6 derived from aspartate. Okay, and also N1. Okay, N1, C4, C5, C6 derived from aspartate. Okay, so basically, if you understand the structure, you should identify from, okay, each component in the structure, what are the sources that contribute, okay, to the build-up of the pyrimidine nucleotide structure. Okay, so now let's look at the biosynthesis of pyrimidine. Okay, so we know that carbamoyl phosphate is the key intermediate that involved in the biosynthesis of pyrimidine. Okay, so now the first step, okay, before that, we should know how carbamoyl phosphate is synthesized. Okay, how carbamoyl phosphate is synthesized. Okay, ingat lagi tak macam mana, what are the reactions involved to synthesize carbamoyl phosphate? Okay, so carbamoyl phosphate is synthesized from the reaction of glutamine and bicarbonate. Okay, check balik. Okay, carbamoyl phosphate is synthesized from the reaction between glutamine, okay, and also bicarbonate. Okay, so this is the reaction is catalyzed by carbamoyl phosphate synthesis 2. Okay, you must be familiar, okay, with this reaction. Okay, kalau you dah belajar urea cycle, if I'm not mistaken, kan? Okay, you have discussed on the urea cycle with Dr. Dina. So you know how this carbamoyl phosphate is synthesized. Okay, then later carbamoyl phosphate is react with aspartate. Okay, it's react with aspartate, so it involves condensation reaction. So this is condensation reaction between aspartate and carbamoyl phosphate, and it catalyzed by aspartate transcarbamylase. Okay, and it produces this carbamoyl aspartate. Okay, if you look, okay, at this reaction, okay, it involves the releasing of inorganic phosphate. Okay, and basically, why does it use carbamoyl phosphate? Basically, it releases carbon phosphate is a very activated molecule, so it does not require any ATP hydrolysis to release inorganic phosphate. Okay, normally kalau kita nak release inorganic phosphate, we need to have this it need to undergo this ATP hydrolysis reaction. But since carbamoyl phosphate is already activated, okay, it's an activated molecule, so it just need to undergo this condensation reaction to release this inorganic phosphate. Okay. Once it produces this carbamoyl aspartate, if you look at the structure of this carbamoyl aspartate, you can see it's nearly to be a closure ring, a closed ring, kan? Nampak tak? Dah nampak dah that ring structure, but it's not closed yet. All right. So the next step, okay, the next reaction, it involves so it involves intramolecular condensation, okay, by the enzyme dihydroorotase, okay, which it involves the removal of water molecule to form this dihydroorotase. Okay, now, so this is where the ring is closed. Okay, so by the removal of water from N-carbamoyl aspartate, which is catalyzed by dihydroorotase, so the pyrimidine ring is closed to form this dihydroorotase. So if the question asks to name the structure of pyrimidine where it form a complete closed ring, so you should know this. This is a dihydroorotase. Okay, so next, okay, what happens to the next reaction? This dihydroorotase, okay, undergo oxidation reaction that catalyzed by dihydroorotase dehydrogenase. Okay, and at this reaction, NAD plus act as a electron acceptor. Okay, and it is from orotate. Okay, so from dihydroorotate, we undergo oxidation reaction to form orotate. It involves an Nd plus as an electron acceptor. Okay, electron acceptor. Okay, so from orotate, okay, from the orotate, okay. So kat sini it already involved this ribose 5-phosphate. Okay, so now we have this orotate. Okay, it's ready now to be attached to ribose 5-phosphate. Okay, so how does it attach to this ribose 5-phosphate? Okay, to form a nucleotide. So orotate react with CRTP, okay, phosphoribosyl phosphate, okay, to form this orotidyllate, okay, to form this orotidyllate, okay, okay, or okay, orotidyllate ataupun OMP, okay, OMP orotidine monophosphate, okay, certain book use orotidyllate, certain book use OMP, okay, orotidine monophosphate, okay, and this reaction catalyzed by orotate phosphoribosyl transferase. So basically, it transfer this phosphoribosyl to the link to the orotate. Okay, and it release the inorganic pyrophosphate. Okay. Okay. So from OMP or orotidyllate, now it is converted to UMP. Okay, tadi orotidine. Now it is converted to UMP or uridylate or uridyl monophosphate. Okay. So this is a decarboxylation reaction that catalyzed by orotidyllate decarboxylase. Okay, orotidyllate decarboxylase. So it's from orotidine to uridyl. Okay, it involves decarboxylation. Okay. Okay, once, okay, because kita kan kalau dalam UMP, kalau dalam pyrimidine, we want to have this uracil ni dalam uridine. Okay, in form of uridine monophosphate or uridine triphosphate. Okay, that's why from OMP, we need to undergo decarboxylation to form UMP. Okay, then from UMP, it is phosphorylated to UTP. Okay, maknanya undergo two phosphorylation reaction to form UTP. Sekejap ya. Alright. So from UMP, from monophosphate, it undergo two phosphorylation steps to form UTP. Okay, and from UTP, it can undergo another reaction, okay, to form CTP. So it has the addition of amino group from glutamine, okay, and it also powered by ATP hydrolysis, okay, to form this CTP, okay, cytidine triphosphate. Okay, so the nitrogen donor is glutamine. Okay, or masters can use ammonium directly. It depends, okay. It depends on certain organism may use glutamine, maybe dekat mammals or maybe plants or bacteria maybe can utilize this ammonia, okay, directly from the atmosphere, okay, to convert this UTP to CTP. Okay, so this is how this is how the biosynthesis of UTP and CTP from carbamoyl aspartate, from carbamoyl phosphate. Okay, ada soalan?
Doktor.
Soalan? Yes.
Doktor.
Ya Fatia?
Step dia banyak sangat lah doktor. Doktor tak ada tips ke?
Step apa yang banyak? Dekat purine lagi banyak. This one is much
Pirin banyak,
simpler.
perimidin pun banyak.
Okay, tak banyak. If you block your mind dengan banyaknya pathway ni. If at the moment kalau you terus block, so if you try to memorize apa, you akan terus block. So now try to open up your heart, open up your mind, and you need to find your own way how you can understand. That's why saya dah bagi tips. I think I already mentioned you. You have to find something that makes you love all this pathway. Macam mana? Okay, satu. Okay, contoh purine lah. Okay, purine. Okay. You need to go back to the structure. Kalau you faham structure, let's say, okay, contoh macam ni. Okay, you dah faham dah. Okay, you have all these steps. Okay, then sambil-sambil you buka ni, you letak structure ni dekat sebelah. So you know that first step involve dekat mana? Formation apa? Which one? Then summarize. Summarize by yourself. That's why to show that you understand, bukan to memorize, okay? First thing first, you need to understand, baru you boleh memorize. Okay, to show that you understand, to show that you know what you read is by to do your own summary. Okay, you need to do your own summary dalam a piece of paper. Okay, that's how I do lah. Okay, itu yang saya buat dulu. Okay, tapi memang sebab I have my affection towards biochem dulu. That's why saya tukar major from microbes to biochem. I already have that. So memang saya tengok pathway, I already okay. It's a very interesting thing for me. Okay, kita tengok pathway macam ni. Because its fact is stated there. Kalau if you just read, you tengok macam ni, you akan rasa banyak. But how to understand every step? Bila if you study the first step, you letak this structure dekat sebelah, so that you understand. Okay, the first step is involve the formation dekat mana? Carbon mana? Carbon 1 of PRPP. Contoh. Bukan, this one involves glutamin. Glutamin dekat mana? Is it carbon 3 or carbon 9? Okay, carbon 9 dulu kan? Okay, because it attached dekat ribose kalau dekat purine. So that you understand that. And identify dekat mana glycine, dekat mana glycine contribute apa? Is it carbon? Okay, glycine or is it? Okay, now dekat glycine side it involves ATP. First thing first, do not block yourself. Okay, open up your heart, open up your mind. Try to understand. Okay, keyword dia kena faham. That's it. Okay, no point to memorize if you don't understand. Okay, maybe kalau you rasa purine banyak, start with pyrimidine. Okay, then you should understand kenapa kita nak, why this pathway of pyrimidine produce CTP and UTP. Why these two? Why not guanine? Why not adenine? Keyword. So you should understand what is pyrimidine. Okay. So pyrimidine dia adalah uracil cytosine. So it produced in the form of this. Kalau nucleotide dia mesti ada, ingat tak? Kalau nucleotide it has all that. Then saya dah go back. Saya start dengan basic structure of nucleotide. So it has ribose. It has that ribose sugar, it has that base, and also it has that phosphate. So this is your nucleotide. So this is nucleotide, okay? Nucleotide, base, phosphate, and also ribose sugar. Okay? So the beauty of biochemistry, the beauty of pathway F is we have all this structure. We can understand what happened in the reaction through the structure. Contoh dekat sini. You tengok, okay? Okay, contoh dekat sini. So what happened? So close, Okay, removal of molecule of water, it cause the ring to close, kan? Okay, so ring closure involve removal of water. Okay, so what happened? Kat mana? Air kat mana yang kat sini? So this H2 dengan O remove as molecule of water, so menyebabkan ring tu tutup. Okay, now next, so what happened to the next step? Oxidation reaction from dihydrotate to orotate. So what happened? Apa berlaku dekat sini? Contoh, okay, you kena tahu. Yes, so dekat sini. So oxidation tu apa? Sebab kita tahu NAD plus is a electron acceptor. So apa elektron yang diterima? So in terms of kat sini, this hydrogen, okay, this two hydrogen is reduced, okay, accept by NAD plus and it reduced to NADH. So you should understand the structure. So that's why saya kata by having pathway is the easiest thing because we have all that structure. You can understand whatever. Kalau you baca macam ni, you tak faham tau kalau kita ada structure. So okay, this structure dihydrotate. So when it undergo oxidations, what happened? So apa berlaku? Apa perbezaan dia? Oxidation tu dekat mana? Okay, so now contoh from orotate to orotate. Okay, so it react with PRPP. So you can now letak PRPP punya structure dekat sebelah. Okay, so look at the name of the enzyme as well. Orotate phosphoribosyl transferase. So it transfer phosphoribosyl. Okay, it transfer this phosphoribosyl and it release this inorganic pyrophosphate. So lepas tu you ambil PRPP punya structure and you look dekat mana this inorganic pyrophosphate yang dia release. Okay, and which part of phosphoribosyl transferase that be linked to this orotate. Okay, if you just read the pathway, you will never understand okay what happened in that reaction. Trust me. Okay, one thing yang saya rasa, okay, biochem lagi senang, contohlah dulu biochem lagi senang daripada microbe. Microbe banyak, bagi saya lah, banyak menghafal. You need to hafal all that nama bacteria, dia sub dia kan, nama kelas dia, general dia lah, apalah, kan. For me, bila ada pathway, you just need to understand. Tengok structure, faham balik. Contoh, glucose phosphate. You dah tahu dah phosphate enzim dia apa? Kan, kalau ada kinase je, it involve transfer of phosphate. So you dah tahu, cuma kena tahu kat mana dia transfer. Dekat carbon 6. Contoh, glucose to glucose 6 phosphate. Contoh. Okay. So, you have that advantage of having pathway and also structure. Semua ada structure. Orotate ada structure. Even you belajar glycolysis system structure, semua ada structure. So that you should understand whatever happen dalam reaction tu saya kata, you jangan boleh depends pada saya punya slide, but you should do your own homework. Okay. Macam saya dulu memang saya bawa textbook pergi kelas. Itu saya lah. Okay, memang saya beli textbook ataupun saya pergi pinjam, saya bawa, then I use textbook to help my understanding. Contohlah. Kalau you baca macam ni, you're banyaknya. No, tak banyak. From here, you boleh identify which step is important. Kat mana important? Of course, one thing you kena tahu, UMP produce dulu or CTP produce dulu satu. And you should know how from UMP we can get CTP. So how from UTP we can get CTP. So you should know it involves this. It involves glutamine and also ETP. Because these two are important because so it tells us about pyrimidine biosynthesis. Kita nak di CTP and ETP. So you understand this. That's it. Mungkin soalan akan fokus on this two reaction probably, or dekat the first one. Alright, ataupun dekat sini. Biasa. Okay, contoh kalau dekat sini kan. Maybe saya boleh pick. Biasanya the first reaction is very important. Okay, the first reaction. Okay, along the way, mana-mana yang melibatkan ATP ke is important. Okay, dekat ring closure pun important. Then you should identify the key metabolite that being produced. Okay, sebab daripada IMP dia akan produce AMP and TMP. So obviously this reaction is very important sebab kita bercakap tentang purine biosynthesis. Okay, boleh tak?
Okay,
That's
doktor.
my simple tips. Okay, you just please don't read. Okay, you can read but with some additional homework. Okay, if you don't utilize all the structure dalam slide, dalam buku teks, you takkan faham. Okay, it does help your understanding. Trust me. All the structure ni, it will help your understanding. Okay, all right. Okay. Okay, so we have complete the pyrimidine biosynthesis. Okay. Next, let's move to the formation of deoxyribonucleotides. Okay, so DNA, okay. Can somebody tell me how DNA is differ from RNA? How DNA differ from RNA? Okay, apart from that, in terms of the structure.
Based on ribose sugar.
Okay, ribose sugar. Dekat carbon mana?
Two.
Okay, so DNA contains deoxyribose at carbon 2, okay, compared to RNA. Okay, and also it contain the base, it contain thymine, okay, compared to kalau RNA dia ada uracil. Okay, so that's the two important difference that makes DNA different from RNA. Alright. Okay, so deoxyribonucleotides are synthesized from ribonucleotides. One thing yang you kena tahulah. Okay, so ribonucleotides are the precursor for deoxyribonucleotides. Okay, okay, can you guys, okay, can we have a break for 5 minutes sekejap? Boleh tak? Before I continue with this slide. Boleh?
Boleh.
Alright, okay. Sekejap Okay. Semua ada ke?
Ya, doktor.
Alright, okay. Alright, okay. So let's go back to the formation of deoxyribonucleotides. Okay, so it derived, okay. So deoxyribonucleotides are synthesized from ribonucleotides. So it's catalyzed by ribonucleotide reductase. Okay, catalyzed by ribonucleotide reductase. So this enzyme, so basically it has several redox, okay, several redox active sulfhydryl groups, okay, sulfhydryl groups, okay, and the enzyme activity is generated through the disulfide interchange, okay, with thioredoxin. So if you look at this diagram, okay, one thing that you need to know that ribonucleotides are synthesized from ribonucleotides. Okay, one. Okay, and what are the enzymes involved is ribonucleotide reductase and also okay. And also, okay, one is the precursor of the deoxyribonucleotide. The second one is an enzyme, okay, which is ribonucleotide reductase. And you need to know that this enzyme is associated, it has all this thioredoxin. Okay, thioredoxin, it also has these sulfhydryl groups, okay, which involved in the formation of deoxyribonucleotides. This sebenarnya banyak lagi in details, okay, but I don't think I'm going to discuss in details on the what happened in the formation on the enzyme activity, because there are three classes of ribonucleotide reductase. Okay, there are three classes. Okay, RNR1, RNR2, and RNR3. And the enzyme mechanism of ribonucleotide reductase basically involve all this disulfide formation, reaction, and also it involves all this thioredoxin. Okay. Because what makes the difference between the three classes of ribonucleotide reductase is each class 1 of ribonucleotide reductase, it contains ferrum, okay, ferrum as a prosthetic group, okay, and I can't remember about RNR class 2, okay, but which kalau RNR class 1 mostly occurs in most in eukaryotes and also aerobic prokaryotes. So RNR class 1 is the most widely used enzymes, okay, dalam eukaryotes. Okay. All right, so now let's move to this tiny delete. Okay, so what is tiny delete? Okay. Okay, thymidylate. Thymidylate or thymidine monophosphate. Okay, deoxythymidine monophosphate. All right. So this DTMP, okay, of DNA. Okay. Okay, kalau ada depan tu deoxy ni, this is a DNA lah. Okay, DTMP is synthesized by methylation of UMP. Okay, methylation of UMP. Alright, take note on that. Okay, daripada DUMP, dia pergi DTMP, it involves methylation reaction that catalyzed by thymidylate synthase. Okay, what does it mean by methylation? They involve addition of methyl group. So daripada mana methyl group tu? Okay. Alright. If you look, okay, if you look at this reaction. Okay, so contoh lah. Okay. Okay, the passes are shown beginning with the reaction catalyzed by ribonucleotide. Okay, if you look, okay, to form, okay, so now from CDP, okay, from RNA, okay, from CDP is converted to deoxycytidine DCDP, catalyzed by ribonucleotide reductase. Okay, same goes to the conversion of UDP to DUDP. Okay, mesti from ribonucleotide to deoxyribonucleotide, okay, it is catalyzed by ribonucleotide reductase. Okay, so from DCDP, okay, to DCTP, it involves nucleoside diphosphate kinase. Okay, it involves phosphorylation. Okay, it involves kinase addition of phosphate group. Okay, from di to triphosphate. Okay. Okay, from DCP to DUTP, it involves deamination. So, you ingat balik tak? Okay, dekat sini, how UTP is converted to CDP by amination reaction, addition of amino group to UTP to form CTP. So, the conversion, kalau nak tukar balik DCTP to DUTP, it involves deamination. It removes that amino group. Okay, and from DUTP to DUMP, it involves reaction that catalyzed by DUTPase. Okay, and from DUMP to DTMP, it catalyzed by thymidine synthase, which it involves methylation yang saya mention tadi. Okay, look at this reaction. Oh, okay. What happened to these two reaction? Siapa yang notice?
Doktor again?
Okay. From DUTP, okay, from deoxyuridine triphosphate, it is converted to DUMP. From triphosphate, uridine triphosphate to deoxyuridine monophosphate. Okay, from DUMP, now it is converted to DTMP. So, what is the, what can you notice from these two reactions?
Phosphate jadi monophosphate.
Okay, so at this step, the first step, okay, before uridine, okay, uridine, before it can be, okay, from UTP, okay, from this UTP, before it can be converted to DTMP, it needs to undergo dephosphorylation, okay, to form DDMP. Once it has only one phosphate, so then it can be converted to DTMP. From uridine to thymidine. Okay, so, okay, if you still remember, okay, in purine biosynthesis, we already discussed on the formation of AMP and GMP, correct? Betul tak? Betul? Ada response.
Betul.
Okay,
Doktor.
ingat tak? Ingat. Okay, do you still remember that in purine biosynthesis we get the formation of AMP and GMP. Okay. Just now we discussed on the biosynthesis of pyrimidine, which involves the formation of CTP and UTP. Betul? So kita belum discuss kat mana macam mana dapatnya thymidine, because thymidine is part of the is part of the pyrimidine. Dia adalah nucleotide. So this is how thymidine is synthesized from DCDP and also DUMP. Okay, jangan you tanya pula DTMP ni apa doktor? No. Okay, because we already discussed nucleotide, we have adenine, guanine, cytosine, thymidine, and also uracil dekat RNA. But uracil and cytosine we have discussed the synthesis dalam pyrimidine tadi. But how can we synthesize thymidine? So this synthesis of thymidylate, okay, or DTMP, is can be synthesized because the thymidine is one nucleotide that is the nucleotide that present dalam DNA kan. That's why kita synthesize dia dalam DTMP. Okay.
Doktor,
Okay.
yang
Yep.
di dalam CDP tu apa? Cysteine.
Cysteine diphosphate. So from cysteine diphosphate, okay. So this is your ribonucleotide. Yang biasa, do you understand the term ribonucleotide, deoxyribonucleotide? This one is your RNA. Okay, So this is your ribonucleotide. Okay, dalam form cytosine diphosphate. Okay. So it undergo reaction catalyzed by ribonucleotide. Kalau ada je ribonucleotide reactors, meaning that we want to convert that molecule into deoxyribonucleotide. So we have this deoxycytidine diphosphate. Okay, so bila dah ada, once it already in the form of deoxy, so we can convert it to DCTP. Okay. And DCTP can be converted to DUTP. Okay, go back to this reaction. Kita tahu UTP converted to CTP by amination from glutamine, addition of amino group daripada glutamine, okay, to form CTP. So kalau kita nak convert balik CTP ke UTP, you need to undergo deamination. Deamination, buang balik that amino group. Remove balik. Okay, and the DUTP, uridine, okay, before we can convert it to thymidine, it need to be converted to DUMP. Kita kena remove dulu that two additional phosphate tu. Okay, by enzyme DUTPase. Okay, from DUMP, then it can convert to DTMP. It can convert to thymidylate to form this that catalyzed by thymidylate synthase to form this DTMP. Okay, dia kalau next reaction ada lagi, DTMP ni dia akan terus convert to DTTP, okay, by phosphorylation reaction, okay, deoxy thymidine triphosphate, okay, biasanya dia akan terus convert, okay, it's going to be phosphorylated to form DTTP, okay, to form DTTP, okay. Okay, if you look at this reaction, okay, DUTP you need to undergo dephosphorylation. Betul? Betul kan? Alright, so now once you have this DUMP, ditukar kepada diethylmidate before it undergo another phosphorylation to form DTTP. Okay. Why does this, kenapa dia kena buang, dia macam, you have this very wasteful reaction. You remove that phosphate, then you undergo another phosphorylation reaction. Why is it like that? Kenapa macam tu? Kan kat sini dephosphorylation, so kat sini after this, you undergo another phosphorylation to form DTTP. Okay. So what happened? Okay, the reason of that reaction, okay, dephosphorylation, then another phosphorylation occurs here, is that cells need to minimize their concentration of DUTP. Okay, kenapa dia kena minimize? Kenapa dia kena undergo dephosphorylation? Sebab cells need to minimize their concentration of DUTP to prevent incorporation of uracil into their DNA. Okay,
Doktor tak dengar.
tak dengar?
Apa tadi
Okay.
yang minimize the
Okay.
concentration of UTP?
Okay, cells need to minimize their concentration of uracil. UTP kan uracil, betul? Uracil kan because kita nak prevent, okay, we want to minimize the DUTP in cells because we want to prevent the incorporation of uracil into DNA. That's why it need to undergo dephosphorylation and convert terus kepada DTMP. Kat sini baru berlaku balik phosphorylation. They want to minimize the concentration. Diorang kena minimize concentration. Otherwise nanti uracil akan incorporate dalam DNA. So kat situ akan jadi haywire lah sistem nanti kan. Sistem kita punya body system. Okay. They must be, yes.
Kenapa tak daripada dioksi tu terus kepada monofosfat je? Kenapa daripada 2 pergi 3 lepas tu jadi 1?
Okay, sekarang ini, okay. Kenapa tak ada specific path? Kenapa why not from here, kita terus CTP tukar kepada DTMP? Why does the formation of DTMP is not incorporated dalam this pathway? Kenapa? Agak-agak ni kenapa? Because thymidine, okay, that's why this slides comes first before saya discuss yang ini. Nampak tak? Because okay, thymidine, okay, semua nucleotide yang lain, adenin ke, guanin ke, apa lagi, cytosine ke, dia present dalam dia boleh dalam form ribonucleotide and also deoxyribonucleotide. But what makes thymidine special? Dia hanya present dalam deoxyribonucleotide. That's why dia tak present kat sini. Nampak tak? Kenapa dia tak involve dalam this biosynthesis? Kenapa from CDP tak terus kat sini? Sebab the CDP, UDP, it need to be converted to deoxyribonucleotide dulu. Sebab tu saya letak slide ni dulu. I want you to understand that ribonucleotide can be converted to deoxyribonucleotide by the action of ribonucleotide reductase. Okay, so you can understand, okay, ribonucleotide is converted to deoxyribonucleotide by the action of ribonucleotide reductase. So once kita dapat dah deoxyribonucleotide from DCDP, it undergo phosphorylation to form DCTP. So once it is in the form of deoxyribonucleotide, so that we can synthesize thymidine or thymidylate in the form of TMP. Faham tak? Apa beza DTMP dengan AMP yang lain? Yang lain, kita synthesize AMP, we synthesize GMP, dia bukan dalam bentuk DAMP. Okay, it's not we synthesize them in the form of DGMP, DUTP. Okay, semua dalam UTP, CTP. Nampak tak? But this thymidine is a very special nucleotide because dia hanya present dalam deoxyribonucleotide. Sebab thymidine, kalau dalam RNA, there is no thymidine. Okay, on uracil yang present kan, in place of thymidine. Can you get the point? Nampak tak? Keyword here, it has this deoxy. It is in the form of deoxyribonucleotide. That's why this slide comes first before the biosynthesis of thymidylate. Because it need to undergo this reaction. Dia dalam deoxyribonucleotide. Okay?
Okay, doktor.
Okay, doktor.
All right. Okay. So I hope you get this. Okay. Keyword here, cells need to minimize the concentration of DUTP because we don't want any incorporation of uracil into DNA. Okay. Okay, one thing that I would like to highlight, okay, nampak tak banyak benda yang saya discuss dalam lecture. Okay, yang tak ada pun dalam slide. So, you should take note sebab kadang-kadang apa yang saya keluar dalam lecture ni akan keluar dalam exam. Okay, that's why I mention to you that slide should be you punya guideline. Okay, other than that, saya akan banyak discuss dalam lecture, dalam kelas. Okay, so take note lah whatever yang saya discuss ni. Alright, okay. Okay, apart from that, one thing yang saya nak highlight is this reaction, this particular reaction. As I mentioned tadi, the conversion of DUMP daripada uridine monophosphate, deoxyuridine monophosphate to DTMP, catalyzed by thymidylate synthase, it involves methylation reaction. Okay, methylation reaction. Okay, so methylation reaction meaning dia mesti ada dia punya donor. Apa sources dia? So donor for this methylation reaction is N5 and10 methylene tetrahydrofolate. Okay, and THF tetrahydrofolate. N5 and 10 methylene tetrahydrofolate. So that molecule act as a donor for methyl, okay, to form this DTMP. Okay, simple je kan kalau kat slide ni. Okay, UMP, DNP by thymidylate synthase. So what happened in that particular reaction? Okay, so bagi saya penting jugalah. Okay, alright. Okay, so that's the end for the biosynthesis reaction. Okay, de novo pathway. Okay, de novo pathway meaning that it's a new biosynthesis pathway lah. Okay, for adenine, we have completed the biosynthesis of adenine, guanine, cytosine, AMP, GMP, CTP, UTP, and also DTMP. Okay. So now let's move to the salvage pathway. Okay, what is salvage pathway for purine and pyrimidine bases? So what is salvage pathway? Salvage pathway meaning that it utilizes all the free purine and pyrimidine bases that release during the degradation activity of nucleotide. Okay, biasanya kalau degradation dia akan release adenine, guanine, okay, hypoxanthine. So the salvage pathway utilize this bases, okay, to produce AMP and also GMP. Okay, this one is salvage pathway for purine. Okay. Alright. Okay, so basically kalau biasanya kalau turnover RNA dia akan release lah this kind of ni lah. Macam saya mention tadi, adenine, guanine. Okay, adenine, guanine, hypoxanthine. Okay, adenine, guanine, hypoxanthine. So in mammals, okay, in mammals, purines are mostly salvaged. Okay, are mostly salvaged by two different enzymes. One is APRT, which is adenine phosphoribosyltransferase. Okay, that mediates the AMP formation using PRPP, okay, from adenine. Okay, so adenine react with PRPP, okay, mediated by APRT, adenine phosphoribosyltransferase, to form AMP. Simple kan? Daripada de novo synthesis nak produce AMP ada 11 steps. Okay. Okay, then and another enzyme which is HGPRT, hypoxanthine, nampak ke? Okay, hypoxanthine guanine phosphoribosyltransferase. So HGPRT. So HPRT it catalyzes the analog reaction for both hypoxanthine and also guanine. Okay, dua-dua ni, hypoxanthine dia akan it react with PRPP to form IMP, okay, by HGPRT enzyme. Okay, same goes to guanine, dia akan react with PRPP, catalyzed by this HGPRT enzyme to form GMP. Okay, so this is HGPRT stand for hypoxanthine guanine phosphorylated transferase. So it catalyzes two different reactions. All right. So what is the importance of this salvage pathway? Okay. Okay, so there is a syndrome called as a Leigh-Nahan syndrome. So this syndrome caused by a severe lacking, severe HGPRT deficiency. Okay, kalau ada a very severe HGPRT deficiency, it causes this kind of syndrome. Okay, so this salvage pathway help to support, producing the hypoxanthine guanine, okay, constantly. Okay, so it's a very important pathway juga, walaupun dia salvage je, but it is a very important pathway. Okay, so what? Okay, so let's look at the syndrome. Okay, Leigh-Nahan syndrome. Okay, so it is a sex-linked congenital defect. Okay, and it affects mostly male. Okay, it affects mostly male. Okay, it results in a very excessive uric acid production. Okay, it results in excessive uric acid production. Okay, and neurological abnormalities. Okay, neurological abnormalities. Okay, neurological function. Okay, okay, apa contoh neurological abnormalities? Okay, dia punya spasticity, mental retardation, and also it can cause a very highly aggressive and disruptive behavior. You tengok gambar ni. Okay, so people who have this kind of syndrome, dia tend untuk bite dia punya finger and lips, and sometimes they tend to injure themselves juga. Okay, because they ada this neurological abnormalities. Okay, dia ada highly disruptive behavior, aggressive behavior. Okay, so dia ada two main important problem with this syndrome. Satu, a very excessive uric acid production, and also neurological abnormalities. Okay, dia tend, gambar dia tunjuk dah. Dia tend to bite dia punya fingers, dia punya lips, okay, and they tend to injure themselves, okay, often. Alright. Okay. What else I would like to share? So the lack of, okay, you can see from this reaction, okay, the lack of HGPRT, dia akan, what will happen if there's a deficiency of this enzyme? Okay, so it will lead to the accumulation of PRPP. So what is PRPP? What is PRPP? So why does this accumulation of PRPP dia akan lead to okay, now, okay, you know that PRPP is one of the intermediate dalam purine biosynthesis. Betul? Okay, so what will happen if this enzyme is lacking? So it will cause the accumulation of PRPP. So the accumulation of PRPP will lead to apa? Okay, go back to the purine biosynthesis, tengok what happened to PRPP. What happened if if we have a very high concentration of PRPP? It will activate one enzyme. Nanti you check balik enzyme apa, aminotransferase something. I can't remember. Okay, so it will activate that enzyme. Okay, so dia akan accelerate the synthesis of purine. So kat sini dia akan banyak berlakunya formation of dia punya degradation kan, akan berlakunya excessive production of uric acid. Okay, if there is an accumulation of PRPP, dia akan activate that enzyme, dia akan accelerate the purine biosynthesis, so it will let, bila ada nucleotide degradation, dia akan ada excessive production of uric acid. Okay, so now you know why is it, I'm sure you tahu kan tentang gout and everything. So the high concentration level of uric acid will cause this gout. Okay, gout selalunya will cause this pain dekat joint, okay, swollen and inflamed joint. It will cause this uric acid, okay. Alright. So it causes this uric acid crystals, okay. Then all this pain pada inflamed joint, it is caused by this uric acid crystals that accumulate, okay, dekat sini lah. Okay. Alright. Okay, barulah now we move to degradation of nucleotide. Okay, we have discussed on the biosynthesis, on the salvage pathway, now move to degradation of nucleotide. Okay, so purine nucleotides are degraded by a pathway in which they lose their phosphate through the action of 5' nucleotidase. Okay. So both AMP and GMP converted to xanthine, and AMP and GMP catabolism yields uric acid as end product by xanthine oxidase. So degradation of nucleotide, so basically we remove our nucleotide as a uric acid, but all this pathway need to be regulated, otherwise we will have a very excessive production of uric acid, so kita akan dapat ni this kind gout lah. Okay, that's why tadi bila kita lacking, that's why salvage pathway is very important, that enzyme HTPRT is very important, because it utilize PRPP, which it react with either guanine, guanine or hypoxanthine to form GMP or XMP. So that's why that enzyme HTPRT is very important. Otherwise, kalau PRPP is accumulate, okay, it will activate that amidotransferase enzyme. Then it will accelerate the purine biosynthesis process. Dia akan, bila ada berlakunya degradation, it will cause the excessive production of uric acid. Okay. Alright, so ini adalah pathway for the catabolism of purine nucleotides. Okay, for guanine, for GMP and MP. So basically, both of these nucleotide, dia akan form xanthine. Okay, xanthine. Basically, dia akan guna enzyme nucleotidase lah, but it will undergo this deamination reaction juga. Okay, so from xanthine, it undergo this xanthine oxidase to form uric acid. Okay, so uric acid is the end product for degradation of nucleic acid. So this one is the catabolism of pyrimidine nucleotides. Okay, from thymine, it undergo reaction to form this dihydrothymine, which later it will produce methyl malonyl semialdehyde, which is going to be further degraded to succinyl-CoA. Bila namanya dah degraded to succinyl-CoA, it will enter. Kalau convert to succinyl-CoA, dia akan masuk mana?
TCA?
TCA cycle, fat cycle. Okay. Nampak tak? Dia ada, there is interconnection between all metabolism, protein ke, lipid ke, it will go back, okay, to your TCA cycle. Dia akan kembali, dia must be some, dia akan produce amino acid, okay, intermediate TCA cycle punya intermediate. Alright, nampak tak dekat sini pun you dah ada amino isobutyrate. Okay, which kat sini it involve aminotransferase, amino group from ketoglutarate to form this methyl malonyl semialdehyde. Okay, you can see, okay, it's involve the the transfer of amino group, okay, from amino isobutyrate to glutamate. Nampak tak? So dah tak ada dah amino group. Amino transfer meaning dia transfer this amino group to glutamate, okay, to alpha ketoglutarate to form glutamate. So now you have this methyl malonyl semialdehyde. Okay. Alright. Okay, so ada soalan? So this is summary, you can just read. Okay, so I think the best summary comes from you. Okay, you should be able to summarize. Kalau you ingat, dia tak susah mana, tak banyak pun. Okay, as long as you understand the pathway, as long as you understand every reaction, so nanti you identify, okay, which reaction is very important. Okay, kadang-kadang saya bukan tanya every reaction pun. Saya tanya, okay, what happened in this catabolism of pyrimidine? Saya adalah tanya, okay, what happened in the first step of meta? Okay, maybe saya tanya kat sini ke, what happened to the end product of catabolism of pyrimidine? So you should know, so what is the end product? Okay, something like that. As long as you can summarize, you understand what happened, that's it. Okay. Alright. Okay. Ada soalan?
Tak ada, doktor.
Okay. Tak ada. Alright. Okay. So we are done with nucleotide metabolism. Okay. We have another half an hour. Okay. After this, okay. I would like you, okay, there is an activity, okay, saya akan attach activity tu kejap lagi kat Putra Blast. Okay, I would like you to, okay, I'll give you two figures, okay, that explain about regulation of purine and pyrimidine biosynthesis. Okay, kita dah belajar pathway kan, every pathway mesti ada regulation. Okay, why is it the regulation process is very important? Kita nak make sure everything occurs smoothly, there is no problem. So we know that, let's say kalau ada problem dengan you ada uric acid, so that we can pinpoint, okay, maybe you have problem with lacking HDPRT, or maybe because of what else? Okay, banyak sebab uric acid, so we can pinpoint. So I'll give you an activity to summarize on the regulation of purine and pyrimidine biosynthesis dekat lid. Boleh?
Okay, doktor.
Okay. Alright. Okay. Nanti I have one simple tutorial lah untuk for this subject. Saya buat dalam crossword puzzle. But I think I would like to wait for you to complete saya punya activity dulu on the regulation ni. Then later baru saya share tutorial lah. Tutorial saya dalam this time dalam crossword puzzle. Okay? Ada soal? Okay, sekejap saya terus masukkan kod. Okay, saya macam mana saya nak buka saya punya tadi macam mana saya nak buka saya punya ni? Macam mana saya nak stop sharing? Tiba-tiba blur. Macam mana saya nak stop sharing ni? Tiba-tiba saya
Doktor cuba tekan dekat share screen, stop sharing.
tak, saya mencari kat situ. Sekarang saya tengah screen sharing. Awak nampak apa kat screen saya? Awak nampak apa kat screen saya?
Nampak Zoom.
Zoom.
Zoom. Okay. So macam mana saya nak stopkan saya punya screen sharing?
Doktor nak share apa?
Tak, saya nak stop sharing je. Meeting controls. Okay, meeting controls. Kat mana saya nak buka? Saya nampak tu saya buat apa ni?
Doktor ada tak kamera kecil kat tepi doktor? Dekat skrin doktor?
Tak ada.
Tak ada alamak.
Tadi juga saya off pula. Cuba Zoom. Jadi gitu saya benda pula apa yang saya buat ni. Okay, sementara lah saya tak jumpa ni kan. So, these are two, nampak tak? Nampak ke? Saya punya.
Nampak.
Okay. So, saya punya, so based on these two figures, nanti saya akan bagi details apa yang saya nak buat lah. Ya, kejap saya mencari saya punya ni. Okay.
Why is the wall? Why is the